Hello students, this week we're going to talk about how we can transfer bacteria from one media to another using aseptic techniques. Microbes are ubiquitous, meaning they are found everywhere. They are found in our environment, for example, in the air, in the soil. You find microbes on surfaces. They can be on your skin, even inside of us. And so basically, microbes are found everywhere, which kind of creates a challenge when we want to protect ourselves against them since they are found everywhere. Now, in a healthcare setting, for example, there are lots of patients that could be vulnerable to infections, and so therefore these patients must be safeguarded from getting exposed to these harmful microbes. And so therefore, it's pretty important for us to discuss aseptic techniques that are used and practiced, used by healthcare professionals to minimize getting exposed to these harmful microbes, or we refer to them as pathogens. Basically, minimize the contamination, right? By these pathogens. Now, these aseptic techniques are not just used by healthcare professionals, but also in a lab setting, especially microbiology lab settings, we also have to follow those aseptic techniques so that students can also protect themselves from getting exposed to the microbes that are used in a lab setting. And so again, when you think of healthcare professionals and how they use those aseptic techniques, for example, you think of how they clean surfaces in between patients. They do not use the same tools between patients. They have to, throw out whatever they're using and go ahead and use new tools when they have a new patient. They wash their hands in between patients. These are examples of some aseptic techniques that they follow. And again, some of these aseptic techniques are definitely shared between healthcare professionals and microbiology students in a microbiology lab setting. So we're going to discuss more about those aseptic techniques in just a minute. Now, what are the goals of aseptic technique? There are two main goals, and those are, number one is we follow those aseptic techniques to protect ourselves, okay? So one of the main reasons we follow those rules is because you want to protect yourself. You don't want to unintentionally get exposed to harmful microbes. And so therefore, again, it's all about protecting yourself. It's also about protecting others that are around you, whether those are your classmates, if you are in a classroom setting, or if you are protecting your patients, if you are in a healthcare setting, if you are in a clinic. Again, you want to make sure that your patients are going to be safe and you're not exposing them, to harmful microbes. You also, if we are in a classroom setting, you want to protect your experiment. You don't want to contaminate your experiment. So those are the two main goals of why we follow aseptic techniques. By the way, if you break the word aseptic, so septic meaning, contamination or basically sepsis. A means without, so without sepsis. So aseptic, you are basically you're following some rules that are going to prevent contamination, okay? Now, what are some examples of aseptic technique? One is in a microbiology lab setting, we usually turn on what we call Bunsen burners. So these Bunsen burners, they are basically going to kind of create a sterilized zone for you. So when you have that open flame, it's going to create a sterilized zone because that open flame, it's going to prevent the microbes in the air from actually falling down on your media plates. And so the whole idea is when you go ahead and have that Bunsen burner on, you want to work close to the Bunsen burner. You don't want to be far away from your Bunsen burner, otherwise you're kind of stepping away from that sterilized zone that you just created. So you always want to work close to the Bunsen burner. You always want to actually feel the heat coming off of the Bunsen burner. That's kind of going to give you, an idea, okay, am I close enough or not? So if you don't feel the heat of the Bunsen burner, then this is kind of an indication that you've stepped far away from that sterilized zone, okay? Another aseptic technique that we follow is washing your hands. And again, this is definitely a shared technique between microbiology students and definitely healthcare professionals. So if we're going to talk about a microbiology lab setting, you definitely have to wash your hands before you even begin your work. And then you must wash your hands after you're done with your lab work. Now, in between, you can wash them as many times as you want. Um, also, gloves, right? So putting on gloves will definitely protect you, especially if you are working with live microbes. Putting on your lab coats, and in a microbiome labs, we request that students put on a lab coat with long sleeves, and it needs to be long enough so that when you sit down, it's going to be protecting your knees. Now, one other aseptic technique that you usually find healthcare professionals doing is, let's say, before they go ahead and inject you, you find that they usually go ahead and sterilize your arm with an alcohol swab. Now, why are they doing that? Because since we said microbes are found everywhere, they're even found on your skin. So that before they go ahead and inject you, they want to make sure that they reduce the amount of bacteria on your skin so that when they go ahead and insert that needle through your skin, you are not going to insert that bacteria on your skin in your blood, right? So it's super important that before injecting any patient, you must you must clean their arms first, or wherever the injection site is going to be, you have to go ahead and clean that skin area with alcohol, or it doesn't have to be alcohol, any other disinfectant would do as well. And then, of course, in a healthcare setting, a physician, a nurse, etc., they cannot, use the same needle or the same tool, between patients. They will have to throw it out, use a clean one. And again, that's going to prevent contamination, okay? So this is an image right here of the virtual lab that you're going to be working on this week in lab. Now, what are some steps that you should take before beginning an experiment? So think about it, okay, some of the aseptic techniques that you should go ahead and make sure that you follow. Number one is you should go ahead and wash your hands, right, before starting your experiment. Make sure that you've put on your lab coat. Make sure you put on your goggles, your gloves, make sure you've tied your hair back. Make sure that you've cleaned, like you've you disinfected your bench area. Okay. Go ahead and um, uh, create that sterilized zone, right, with your Bunsen burner. Okay, so as we said, the Bunsen burner, they're basically used to create that sterile zone. So you want to make sure, as I said, when you're working, work near your Bunsen burner so that you can stay within that sterilized zone, because the heat, okay, from that open flame, it's going to kind of create a zone around that Bunsen burner. It's basically going to prevent the bacteria from the air from falling down onto, let's say, your media plates, okay? And so it's going to prevent that from happening. And so that's why it's important that you go ahead and you work near that Bunsen burner. Now, other than creating a sterilized zone using the Bunsen burner, what are Bunsen burners used for? They're also used to sterilize your equipment, the tools that you're about to use. And so, for example, this is a tool that we use a whole lot in a microbiology lab, and this is the inoculating loop. Now, inoculating loops, first of all, what are they used for? Inoculating loops, they're used to transfer a loop full of bacteria. Okay, so we use them to grab basically a sample of a bacteria. So what do we call a sample of a bacteria? We call it an inoculum. Okay, and they look something like this. This part, it's made out of metal. So before you use that inoculating loop, we got to make sure that it's sterile. So how do we do that? We go ahead and we pass it through the flame. Now, this is not a quick thing to do, like quickly pass it through the flame. No, you got to take your time, take that loop, pass it through the flame until the loop turns orange red. Okay, got to make sure that this whole entire part, okay, not the part you're holding, okay, because the part that you're grabbing, it's it has a plastic coating, so you don't want to, of course, pass that part, otherwise it will melt. You only want to pass the metal part, okay? So you pass the whole entire metal part of your inoculating loop into the flame, as I said, until it turns red, okay? Once it turns red, um, don't use it right away. Otherwise, you're going to kill your bacteria, because right now it's going to be like, very hot. So, you just to make sure, after you go ahead and you sterilize your loop, just wait, okay, just a few seconds before you go ahead and grab a sample of that bacteria, okay? Another thing, by the way, about the Bunsen burner, the flame, make sure that the flame is kind of like, there's a part of that flame that's like blue, okay? That's going to be like the hottest part, and so that's what you, again, want to pass your flame into, that kind of blue part of the flame. And then, of course, like, why is there a Sharpie there? Because you got to make sure that you label everything, right? And this is pretty important, because you want to make sure that you're labeling everything. You're labeling your tubes, your plates, whatever you're using, so that what is this, what's on that plate, what's in the tube, etc. Okay, so make sure that you always label your plates and tubes. Now this right here, you have a tube that has some liquid medium, and the liquid medium is known as a broth. So broth is a liquid medium that doesn't have any agar in it, okay? And you can see right here, it's labeled E. coli. So this is a liquid medium with cultured E. coli, meaning growth, like you have growth, you have E. coli growth in that liquid medium. Now, when you go ahead and open up that tube, make sure again that you are pretty close to the sterilized zone. And then what we do is when you go ahead and uncap that tube, never place the tube on the surface. Otherwise, you're going to contaminate it. So when you uncap it, make sure that you hold on to that cap, okay, in between your fingers. Then what do? We grab this tube and you actually get it close to the open flame and you flame the mouth of your tube. Okay, so you flame the mouth of your tube, and again, this prevents contamination. This prevents any microbes in the air from entering into your tube. Okay, and then before you cap it, okay, so let's say you uncapped it. After sterilizing your loop, you went ahead and you dip your loop into the liquid broth so that you can get a sample of the E. coli in that tube. Now, should you cap it right after? No. Before you go ahead and cap it, make sure that you flame the mouth of the tube one more time. And then you're ready to go ahead and cap it. Those are some tools that we use, okay, in a microbiome setting. So we talked about the Bunsen burner, and we talked about how the Bunsen burner, it's important because it creates that sterilized zone for you, and you always have to work close to the, close to the Bunsen burner so that you can always stay in that sterilized zone. We also said how the Bunsen burner is used, right, to sterilize the tools that you're about to use, okay, such as the inoculating loop. Okay, so before you use the inoculating loop, you must pass it through that Bunsen burner flame until it turns red. Okay, so again, you keep putting it in there until this whole entire part turns red. Okay, but as I said, after it turns red, just make sure that you wait a few seconds before you actually use it. Otherwise, you're going to kill your bacteria. Now, the inoculating loop looks something like this. It has a loop on, at the tip of it. And we use the inoculating loop to basically grab samples of bacteria. Okay, so anytime you want to, grab an inoculum, okay, like a sample of bacteria, you can use a loop. Can you use other stuff? Yes, you can, but this is one of the tools that we go ahead and we use. Another one that we use is what we call a needle, okay? Now, a needle looks something like this, and again, before using that needle, again, you have to sterilize it, so we pass it also, into that, through that flame, and again, make sure that it this whole entire part right here turns red. Wait for a few seconds before you use it so that you don't kill your bacteria. And we usually, we use that tip right here, which is sharp, okay? But we use it so that we can go ahead and take a sample of our bacteria. Now, we usually use needles to stab a type of media that we call deeps, okay? I'll show you a picture of a deep, but just to introduce you to a deep is basically a type of media that does have agar, but not too much agar, okay? So a deep, it's not going to be a solid medium, it's going to be semi-solid, okay? And so the needle, we usually go ahead and use it with the deep, and you basically stab your deep, and I'll show you how to do it. A loop is usually used when you want to transfer a bacteria into a broth medium or onto a plate or onto a slant, and I'm going to show you what's a slant, okay? So those are different physical forms of media. Remember how we talked about the different chemical forms of media? When we talked about how we have a standard media, how we have selective media, we have enriched media. So those are different chemical forms of media where you can add, special nutrients, and so therefore you can go ahead and change the chemical form, okay, of the media. Right here, we're not talking about the chemical form of the media, we're talking about the physical form, okay? So we talked about plates, okay, and we said that you can pour your media onto these, onto these dishes, and then you get to what we call a plate. And this type of medium, it's solid, okay? It does have agar in it, and the medium, it's basically solid, and it acts as, a good large surface area for you to go ahead and inoculate that surface, meaning introduce the bacteria that you want to grow onto that surface. So this is the plate right here. Now another physical form of a media is what we call a deep, okay? And a deep is basically a tube right here that we fill with the medium. Now it does have agar, but not too much, and so the deep ends up being a semi-solid, okay, type of medium. The agar is solid, okay, the deep is semi-solid. So if you want to inoculate the deep, the best tool to use is the needle. So you basically take your needle and you literally stab, okay, you literally stab the deep, okay? That needle is going to have the bacteria on its tip, and so when you stab the deep, you're basically going to be introducing the bacteria into the medium, okay? We have a slant. A slant is, again, we pour it in a tube. Now the slant does have agar, okay, and it's going to be solid, okay. So a slant is a solid type of medium. When you pour the medium, you go ahead and you tilt your tube, okay? And so when the agar basically solidifies, since you had the tube slanted, you get this nice slant right here. To inoculate the slant, we usually use an inoculating loop, okay? So let me kind of, show you. Let me grab my pen. Okay, so when you go ahead and you inoculate the slant, we usually use the inoculating loop, and you kind of create like a zigzag form right here on the slant surface. Okay, so you usually use the inoculating loop and you kind of create that zigzag pattern. Okay, with the deep, as I said, you use the needle and you stab it. Okay, so you introduce it by stabbing it. The plate, you use the inoculating loop, and again, you create that zigzag format like so. Okay. I mean, this is one way of, inoculating a plate. There are different ways of how you can kind of create that zigzag pattern. We're going to talk about it later, but this is one way you kind of create that zigzag pattern throughout your whole entire plate. Now, a broth has 0 agar. Okay, so the broth is liquid. It has 0 agar, and so it's completely liquid. We usually use an inoculating loop, okay, when you want to inoculate your broth. So you basically take your inoculating loop and you kind of, again, just stick your, basically like dip your inoculating loop in there, and usually kind of flicker the broth tube or shake it a bit just to kind of make sure that, whatever was on the tip of your inoculating loop just kind of, got mixed throughout your broth. Okay, so those are the different physical forms of media that we have. Okay, either it comes in a completely liquid form, we call it broth, a solid form, but we tilt it, so we call it a slant, a semi-solid, and we call it deep, or solid, and we pour it in those dishes, so we call it a plate. So slants, deeps, and plates do have agar. Okay, broth has 0 agar. Okay, so one more time, the physical forms, we said we have liquid, okay, we have semi-solid, and we have solid. So a liquid type of media has 0 agar, and that's why it's liquid, because remember the agar is going to be your solidifying agent, okay? So without adding that agar, without adding that solidifying agent, your medium stays liquid. An example of a liquid medium, the broth that we just talked about. Now, what is the broth used for? The broth is used when you want to grow large quantities of bacteria. So if I want to grow a lot of bacteria, we usually use broth, okay? Now, a semi-solid type of medium does have agar, okay? Usually 0.5 to 0.7% of agar, okay? An example of a semi-solid is the deep, okay, that we just talked about. And what are deeps used for? Well, deeps are usually used for testing the oxygen requirement of your bacteria. And I'm going to explain that in a minute. And they're also used for motility. You want to know whether your bacteria is capable of moving, okay, or whether your bacteria doesn't really move, okay? Remember how we said bacteria can move around, for example, if they have flagella, for example, so if they have flagella, they're going to be able to move around, so they're going to be motile. If they don't, have flagella or any other forms of, structures that can help them move, then they're non-motile. So a deep can actually help you determine that, and I'm going to show you how. A solid medium does have agar. So if you compare, you will see that solid medium, they have the highest percentage of agar, followed by the semi-solid, and then of course, the liquid has 0. What's an example of a solid medium? I showed you 2, right? The slant and the plates. Okay. Now, what are they used for? So slants are good for transporting bacteria. So let's say I'm working in a lab and I want to transfer my bacteria to another location. Okay, let's say I'm working in a lab in Glendora, and I want to take my my bacteria because I want to go to a lab that's in Pasadena. Okay, so I want to transport my bacteria in between labs. So it's best to transport your bacteria, using the slants. Okay. Slants are are also good for storing your bacteria. Okay. Slants, they are they come in right in tubes. I showed you the picture, and they're capped. So actually, those caps are going to prevent lots of contaminants from entering into your tubes, and so therefore, they don't get contaminated as easily. And so they're good to store your bacteria for, longer periods of time. And the plates are used to quantify the bacteria because you get those colonies on your plates. So you're going to be able to quantify the number of colonies. You're also capable of isolating your bacteria. And we're going to talk about, how I can isolate my bacteria. You can basically like pick a certain colony and go ahead and grow it on another plate, and therefore you can isolate your bacteria this way. Now, the tools that we use, okay, when you are inoculating a broth, we usually use an inoculating loop. When you are inoculating a deep, we usually use the needle, okay, so you basically like stab your deep. When you are inoculating your slants or your plates, again, we usually use the inoculating loops. Now you have notes that are provided for you on Canvas about the aseptic technique. So go ahead and kind of take a look at it. It kind of explains how you should, as I said, like when you're using a needle or a loop, you got to pass it through the flame so that you sterilize it. How when you uncap a test tube, you got to make sure that you flame the mouth. So again, kind of goes over that with images. So go ahead and take a look at it. And then there are a bunch of videos that are also, helpful. So go ahead and take a look at them as well. And then, of course, you have the virtual labs on Connect that will help you out as well. Okay, now let's take a look at a bunch of images of some bacterial growths that you can get in a broth. So when you have a sterile broth, meaning you don't have any growth in that broth, and so therefore it's sterile, it's going to look something like this, and it's going to look clear. And the light is going to be able to shine, through your broth, and that means you have absolutely no growth in it. Now, if you inoculate that sterile broth, right? So if you grab your inoculating loop, after sterilizing your inoculating loop, of course, you grab the sample of that bacteria, and you go ahead and you inoculate your broth. We usually leave that inoculated broth in the incubator from, 24 to 48 hours. That's usually how long it takes, before you see any growth. Now, when you see growth in your broth, you're going to know that you do have growth in your broth because your broth is going to turn cloudy. Now, what's the term for cloudy that we use? We use the term turbidity. So turbidity is basically cloudiness. Now, the degree of the turbidity can vary depending on how much bacteria is growing in your broth. So if the turbidity level is low, that kind of tells you that, okay, you do have bacteria growth, but not a whole lot, so it's low growth. If the turbidity is medium or moderate, that means you do have more bacteria growing. If it's high, okay, and your broth is really turbid, really cloudy, then again, it tells you that you have more bacteria growth. So again, the level of cloudiness can vary, and that will be an indication of your bacterial growth. Okay, this link right here, if you guys click on it and scroll all the way to the bottom of the web page, you are going to see, images of low, moderate, and high turbidity growth in a broth. So there are some other characteristics other than the turbidity that you can see in a broth. Okay, so in addition to turbidity, okay, and you guys see how I said in addition? So turbidity is one main thing that you will always have if you have bacterial growth. So if you have bacterial growth, you're always going to, your broth is going to turn turbid. Okay, now again, the level of turbidity might vary, but if you have bacterial growth, you must have turbidity. Now, on top of the turbidity, you might see something like this. You might see this film, okay, like a slimy film on the top of your broth, and we call that a pellicle. Okay, now, do you always have to have a pellicle? No, you don't have to. Okay, it's not a must. And then sometimes you're going to see growth at the bottom of your tube, and that's called a sediment. Okay, so sometimes when your bacteria grow, they're pretty heavy, and so they kind of settle down to the bottom of the tube, and we call that a sediment. Now, can you have both a sediment and a pellicle? Yes, you can. I mean, right here, do you guys kind of see that at the bottom of the tube? So yes, you can have both sediment and pellicle. Okay, or your broth is just going to be turbid and that's it. You're not going to see a pellicle, you're not going to see a sediment, so again, it's not a must. Can you have a pellicle without turbidity? Absolutely not. Can you have sediment without turbidity? No. So again, a pellicle and a sediment are in addition to turbidity. You will never find them just alone because again, a pellicle, for you to have a pellicle, that means you have some bacteria growth in your broth. And so again, the broth is going to turn turbid. And again, for you to have sediment, again, that can happen if you have bacterial growth. So again, you must see the turbidity, so you will never see sediment on its own or pellicle on its own. Okay, that will be in addition to the turbidity. This is a slant right here. Okay, so remember a slant, it's a solid medium. And again, we usually slant it so that as the agar solidifies, you get this slant right here. And again, this is the surface part that gets inoculated with the bacteria. So we usually go ahead and we use an inoculating loop, and you inoculate the surface right here, kind of like in a zigzag pattern. You stick it in the incubator, you leave it for 24 to 48 hours before you can start seeing growth. Now, if you go ahead and take a look at, you have two slants, take a look. Notice how you can see growth on both of them right here, but do you notice something different about them? I mean, one hint is the color, right? So this kind of tells you that there are two different bacterial species growing in this slant tube versus that one. So it turns out that there are some bacteria, when they grow, they release a certain pigment, okay? So some bacteria, they're gonna look white, okay? Some of them are gonna look pink. That's the pigment that they produce. Now, you might wonder, well, if I see bacteria growing on my slant and it's pink on the slant, if I go ahead and inoculate the broth using the same bacterial species, is my broth gonna give off a pink color? Yes. Okay, what if I, again, inoculate my plate using the same bacterial species that's growing on this slant? Am I going to see pink growth on my plate? Yes. Okay, so that's, again, if you're using the same bacterial species. it is going to show off the same exact color. It's not like, oh, okay, it's gonna only show the pink color on a slant, but you're not gonna see it on the plate. No, you're gonna see it in, in both. Maybe in the broth, it's gonna be a bit hard, especially if you don't have like too much growth, okay? But I mean, you're still gonna see that pink color, for example, throughout the different physical forms of the media that you use. Okay, now remember the deep? We talked about the deep and we said that the deep, it's a semi-solid, so it does have agar, but not a lot, so that's why it's a semi-solid type of medium. And we said that if you want to inoculate your deep, what do? We use a needle. And as I said, you literally stab the needle, okay, down into your deep. Now, the deep, as I said, it's used for two things, right? One, it can help you figure out the oxygen requirement of your bacteria. Two, it can help you determine whether your bacteria is motile or non-motile. So let's talk about the oxygen requirement first. How am I going to know what is the oxygen requirement of my bacteria? You can determine that after you stab your deep and after you've left it, to in the incubator for about 24 to 48 hours. You come and take a look at where is your bacteria growing, okay, in your deep. So if you see something like this, where most of your growth is at the very top of your deep, that means that your bacteria is what we call obligate aerobe. Obligate meaning a must, aerobe meaning air, okay, oxygen. So obligate aerobe means that your bacteria must have oxygen for them to grow, okay? It's a must, okay? Not optional, it's a must that they have oxygen for them to grow. And so you're going to notice that obligate aerobes, they're usually going to grow at the very top of your deep test tube. Why? Because this is where you're going to have the most oxygen because it's closest to the top, right? So you're going to see them growing up here. Okay, well, what if I see my growth throughout the tube? If you see the growth throughout the tube, that means that your bacteria is what we call facultative anaerobe. Facultative anaerobe are bacteria that can grow with or without oxygen, okay? So if you don't provide them oxygen, they're going to grow. If you provide them oxygen, they're also going to grow. So they can grow in either condition. But do they have a preference? Yes, they prefer oxygen. Okay, guys, so facultative anaerobe, they prefer oxygen, but if there is no oxygen, they're still going to grow. Okay, so you're going to see that they grow throughout the tube, but you're going to notice more growth towards the top. Even though you're going to see it throughout the tube, more growth should be at the top. Why? Because they still prefer oxygen. Okay, guys? Now, what if you see the growth at the bottom of the tube? Now, if you see the growth only at the bottom, okay, then we call your bacteria obligate anaerobe. So obligate meaning a must, anaerobe means without oxygen. So it's a must for these bacteria to not be exposed to oxygen. Those bacteria, oxygen for them is actually poisonous. We're going to explain why later. But they cannot tolerate oxygen at all. It's poisonous for them. And so you're going to notice that they try to steer away from the oxygen. So therefore, they go all the way down to the bottom of the tube where there is no oxygen, and you will find them growing down there. So do you see how a deep can help you determine the oxygen requirement for your bacteria? How can we figure it out? Depending on the pattern of the growth that you get. And so therefore, it is pretty important that when you handle your deep, especially after incubation, do not shake the deep tubes, okay? If you shake it, you're going to mess up the pattern, okay? And so your data is all going to be messed up. So make sure that after you incubate your deep, you handle it very carefully and do not shake those tubes. Now, we also said a deep has another purpose, and the other purpose of the deep that are used, we use them for, is to determine the bacteria motility. We want to figure out whether our bacteria is motile or not, okay? Now, again, as we said, with a deep, you go ahead and you stab it. Now, notice right here, after you've stabbed your bacteria, how you see this red color throughout the tube. That means that your bacteria is motile. Notice how here, when you stabbed it, you don't see that red color throughout the tube. You only see it right here where you stabbed it. That means your bacteria is non-motile. Now, so again, here it's motile, and here it is non-motile. So first of all, let me kind of explain something. Now, is this red color produced by the bacteria? In this case, no, because this is the deep that we used. It's called TTC deep. Now, remember how I said some bacteria, they naturally produce a pigment? Okay, kind of like that slant picture that I showed you with the pink growth on it. So some bacteria naturally produce pigments, okay? If they naturally produce pigments, well, okay, that's going to be easy to figure out their motility because you're going to see their color, okay? And if you see their color spread throughout the tube, you know it's motile. If not, then they're non-motile. But the problem comes with the bacteria that do not produce any pigments. So if they don't produce any pigments, how am I supposed to know whether they're motile or not? Again, if they don't produce any pigments. And so we use a deep known as TTC deep. TTC, you guys, it's a dye. It's a colorless dye, okay? Now, when the bacteria grows, they usually break down that dye that's found in this deep, and when it breaks it down, it releases a red color, okay? So even if your bacteria doesn't produce any pigments, no problem, because as the bacteria grows in the TTC, it breaks that colorless TTC dye, and as it breaks it down, it turns it into a red color. And so if your bacteria is motile, as you guys can see, it's going to spread, right? Your bacteria growth, okay, it's going to grow. That bacteria is going to grow throughout the tube. And as it's growing throughout the tube, what is it doing to that colorless dye? Well, it's breaking it down and it's turning, kind of produces that red color, okay? But if your bacteria is non-motile, the dye, that TTC dye is only going to be broken down right here where you've stabbed your deep, and so you're only going to see that red color right here, okay? No matter what type of bacteria, by the way, grows in the TTC deep, you're gonna see that red color, because again, the TTC, when it breaks down, you get that red color. So you look at your tubes. If you see that red color throughout, that means your bacteria is moving throughout the whole entire tube and they're motile. If not, then they are non-motile. Okay, so you're going to be given pictures like this. You have your deep. You can tell that this is a deep because you can see where the needle, okay, where you stabbed it. Here's the slant, here's the broth. So you're going to take a look at the growth patterns, and you're going to answer a few questions on your worksheet. And then right here, you have another, set of images of Bacillus subtilis. And again, you have the broth, the slant, and, the deep. And then here you have Pseudomonas aeruginosa. And again, you want to go ahead and take a look at these images right here of your broth, and you're going to also answer a few questions on your worksheet. Right here, you have two images of the deep for the bacteria Staphylococcus aureus. Again, those are TTC deeps. Actually, this one is Staphylococcus aureus. This one is Pseudomonas aeruginosa. And so based on their pattern of growth in the deep, you should be able to determine whether this bacteria is motile or non-motile, and again, whether Pseudomonas aeruginosa is motile or non-motile based on the growth pattern.